Now let us see how many round green phenotype was obtained by the Punnett square. So round green means it should have either one capital R or two capital R's, and it should have small y. Only then the green color will be observed. So let us see it. So we are able to find capital R here, so it codes for roundness, and small y. So again it codes for the green color. So let me change the color to green here. Okay, so I'm writing as number 1 here. So 1, the phenotype which has round green is 1 now. And then coming to the next one, this is capital R, small y. Again it will code for round green because it has one capital R and two small y. So it is 2. Okay, and then let us find another seed which will have the same characters. Okay, so we don't have round green here. Here also we can't find round green, and here again we can find round green because one is capital R and other two are small y. So again it codes for round green. So it is number 3. So how many round greens are formed? So it forms around 3 round green. Okay, so 3 round greens are formed. Now let us coming to the next phenotype, which is the wrinkled yellow. Let us find how many wrinkled yellows there. Let me change the color to red. So let us see the wrinkled yellow. So how should be this wrinkled yellow? Wrinkled, to denote the wrinkled, it should have small r, and to have this yellow, either one capital Y should be there or else two capital Y should be there, right? So let us see, here we have finished the phenotype and see this. So it is showing small r, which codes for wrinkledness, and then it has two capital Y also. So it codes for wrinkled yellow. This is number 1. And again the next one also, small r, capital Y, small y. Again it codes for wrinkled yellow. So it is number 2. And then small r, capital Y, small y. Again it codes for wrinkled yellow. So how many wrinkled yellow will be there? There will be 3 wrinkled yellow, right? So 3. Now let us see the wrinkled green. So wrinkled green is just 1 in number. We are able to find only 1 wrinkled green here. So how many wrinkled green is there? It is just 1. Let me write as 1 here. So what is the ratio now, children? So the ratio of the phenotype of the dihybrid cross will be 9:3:1. Okay. So what is the phenotypic ratio, children? It is 9:3:3:1. This is the phenotypic ratio. And as Pendle said, when he cross pollinated the round and the wrinkled, and same way when he cross pollinated yellow and the green colored seed, he got the phenotypic ratio of 3:1, right? So 3 among the 4 were yellow and 1 was green, and same way in round wrinkled also, 3 of them were round and 1 were wrinkled, which means that 75% of them were showing the dominant trait and only 25% showed the recessive trait, right? So we can expand this 3:1 as 9:3:3:1, right? So എവിടുന്ന ഈ 3:1 വരുന്നതെന്ന് മിസ് പറയുന്നത് മനസ്സിലാവുന്നില്ലേ? റൗണ്ട് ജസ്റ്റ് ഒരു മോണോ ഹൈബ്രിഡ് ക്രോസിൽ റൗണ്ട് ആൻഡ് wrinkled മാത്രം ക്രോസ് ചെയ്യുമ്പോൾ they will get 3:1, right? And same way yellow and green alone, if it is crossed, it will give rise to 3:1, right? So these these two 3:1 can be derived in this way. Okay, so it is 9:3:3:1. Clear, children? Does this 9:3:3:1 ratio show? It shows that there was round yellow. 9 of them among the 16 were round yellow, and 3 among the 16 were round green, and 3 among the 16 was wrinkled yellow, and 1 among the 16 were wrinkled green. So totally 16 possible genotypes are there and gives rise to a ratio of 9:3:3:1 phenotype. right? And if you want to calculate this genotype, it is different from the phenotype. It is completely different from the phenotype. Okay, so let us see the law first, and then we will see what are the phenotypes possible from this. So this is how we can represent the round yellow and wrinkled green are combined to form the capital R, small r, capital Y, small y, which is round yellow again, and it produces the possible gametes which are capital R, capital Y, capital R, small r, capital Y, small r, small y. So these are the possible gametes of both the parent, and when they were self-pollinated, we are able to find such a phenotypic ratio which is 9:3:3:1. So it is derived as a combination series of 3 yellow:1 green with 3 round:1 wrinkled. So this was the phenotypic ratio in the monohybrid, and this can be derived in such a way, that is 9:3:3:1. Right. So now let us see the law of the dihybrid. Before that, let us see the phenotype, that is genotypic ratio. We saw the phenotypic ratio. This is the genotypic ratio. So each one of the genotype which we see here is different. We can't commonly write the ratio here because many new genotypes has been formed, right? So each one has to be written separately. So there is one capital R, one capital Y, right? So this one is 1. So it is written as 1. Okay, capital R, capital Y. Now comes the capital R, capital Y, small y. So how many are there in such a genotype? So this is 1, capital R, capital Y, small y. And same way, we are able to find another one which has the same genotype. So it is 2, right? And then comes the so this capital R, small r, capital Y, it is here, and also it is found here. So capital R, small r, capital Y, capital Y. So this genotype is 2 in number. So if you see the other, can we find any other same genotype? No, it is not there, right? So only 2 of them are capital R, small r, capital Y. Now coming to the next one, which is capital R, small r, capital Y, small y. Let us see how many are them. How many are there like this. So capital R, small r, capital Y, small y. So one we have got it here, and then capital R, small r, small y. Again it is here, and then capital, this won't be there, this this is different, and here capital R, small r, capital Y, small y again. Okay, now we have got 3, and then we have another one which is capital R, small r, capital Y, small y, right? So other those, they don't have this genotype. So all these 3 genotypes are capital R, all these 4 genotypes are having capital R, small r, capital Y, small y. So there are 4 in number. And next one, you can see after that it forms the capital R, small y. So how many are there? 2 number are there, that is this one, capital R, small y, and here also we have capital R, small y. So there's 2 in number, and then comes small r, capital Y. So this one, how many are there? It has small r, capital Y, it is just 1 in number. So the ratio is written as 1, and then small r, capital Y, small y. So it is also 2 in number here and here, right? So small r, capital Y, small y. 2 numbers are there, so the ratio is 2 again. And then comes the small r, small y, it is 1. So what is the genotype, children? Genotype ratio will be 1:2, 2:1:1:2. That is, it forms the 1:2:1:2, 2:4:2:1:2. The possible genotype ratio, children, it is 1:2:1:2, 4:2:4:2:1:2:1, right? So you can derive it with the help of the Punnett square, right? So this is about the dihybrid F2 generations, genotypic ratio and phenotypic ratio, right? So now let us see the law of independent assortment. So it is based on the results of dihybrid crosses. He came up with the law of independent assortment, and it states that when two pairs of traits are combined in a hybrid. What are these two pairs of traits? Two pairs of traits are nothing but characters which we took, round, wrinkled, and the yellow and green. So when two pairs of traits are combined in a hybrid, the segregation of one pair of the character is independent of the other pair of the character. So if you have taken round yellow, this round and yellow are not working together, they are separating out, they get segregated and they become independent and they carry out this gamete formation process. So when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair, right? So one pair is very independent from the other pair of the character, right? So this is how they occur in the living organisms. Clear? From the Punnett square, we can effectively understand the independent segregation of two pairs of genes during meiosis and the production of egg and pollen in the F1, that is capital R small y plant, and consider the segregation of one pair of gene, that is capital R and small r. 50% of the gametes have the gene capital R and other 50% have small r. Now, besides each gamete, either capital R or small r, it should also have the allele capital Y or small y. And the important thing to remember here is 50% of segregation of capital R and 50% segregation of small r is independent from 50% segregation of capital Y and 50% segregation of small y. That is small y. Therefore, 50% of small r bearing gametes has Y and the other 50% has small y. Similarly, 50% of capital R bearing gametes Y and the other 50% has small y. Thus, there are 4 genotypes of gametes which are capital R, capital Y, capital R, small r, capital Y and small r, small y. And it has a frequency of 25% or 1/4 of the total gamete produced. So, they are talking about the gamete formed here, right? So, the possible gametes which are formed. So, this capital R has 50% chance to obtain capital Y and same way they have other 50% chance to obtain small y. And same way, the small r will have 50% chance to obtain the capital Y and other 50% chance to obtain the small y. So, they are saying how the gametes are produced. So, the ratios, that is the percentage is 25%. So, each of these alleles will have 25% chance to reach a single gamete, right? So, this is how the dihybrid cross works in the Mendel's experiment, and this is what is the definition for law of independent assortment. So, when two pairs of traits are combined in a hybrid, segregation of one pair of the characters independent of the other pair of the characters. That is why they are independently working and they are giving rise to new characters also. Clear, children? Okay. So, now let us move on to the next part, which is the chromosomal theory of inheritance.